Welcome back. When we culture Escherichia coli in the lab, we keep it precisely at 37 degrees Celsius. Why? Because E. coli is a mesophile. Its molecular machinery, its enzymes, its membranes, evolved to function optimally in a narrow middle range. But the vast majority of the biosphere is not middle range. It is cold, highly acidic, alkaline, or under crushing pressure. For decades, biology assumed these environments were sterile. We were wrong. Bacteria and their cousins the archaea have occupied these extreme niches for billions of years. In fact, the earliest life on Earth likely was thermophilic. Today, we will ask, why? We won't just list the weird places they live. We will dissect the mechanisms, the biochemical and thermodynamic strategies that allow a single-celled organism to define the absolute limits of life. Adaptation to environmental stress requires systemic changes. An organism cannot simply protect one pathway. It must re-engineer its entire physiology. Let's focus on two fundamental cellular requirements: keeping the cytoplasm stable and keeping the membrane intact. Cellular and biochemical adaptations. Thermophiles, heat and stability. Heat is the ultimate disruptor. It increases kinetic energy, causing biological molecules to vibrate violently. For a mesophile, high heat means protein denaturation, unfolding, and membrane lysis. The membrane becomes too fluid and essentially disintegrates. Thermophilic bacteria solve this thermodynamically. First, they modify their membrane composition. Mesophiles rely on unsaturated fatty acids, think olive oil. Thermophiles incorporate long-chain, highly saturated fatty acids, which pack tightly together. Many also utilize hapenoids, steroid-like molecules that reinforce membrane rigidity. The resulting structure is far less sensitive to temperature-induced fluidity. Psychrophiles, cold and fluidity. The challenge of the cold is the opposite. Molecular motion slows down. Enzymes become rigid, and membranes freeze, halting transport. Psychrophiles adapt by increasing membrane fluidity. Their lipids have high concentrations of unsaturated fatty acids, creating, quote, kinks in the tails that prevent tight packing, keeping the membrane pliable. Thermophilic enzymes are often stabilized by increased numbers of ionic bonds, salt bridges, and a tightly packed hydrophobic core, minimizing exposure to the denaturing solvent. Stability, ions, and pressure. We can now drill down to specific chemical environments. Halophiles, the Salton problem. Halophiles live in environments where the external sodium chloride concentration is approaching saturation, for example 5 molar. For most organisms, this high osmotic pressure causes severe dehydration, plasmolysis, as water rushes out to balance the solute concentration. Halophilic bacteria use two strategies. The first, less common in bacteria, is the synthesis of organic, compatible solutes, like betaine, that balance osmotic pressure without interfering with metabolism. The second strategy, the quote-quote Salton approach, involves accumulating massive internal concentrations of potassium ions to balance the external sodium ions. This is an energetic investment. To function in this high potassium cytoplasm, the entire proteome must adapt. Halophilic proteins possess a high percentage of acidic amino acids, aspartate, glutamate. These negatively charged residues require a high salt cloud like potassium to shield the charge and stabilize the folded, active protein conformation. Acidophiles and alkaliphiles, the pH barrier. Maintaining cytoplasmic pH near neutrality, pH 6.5 to 7.5, is non-negotiable for bioenergetics. Acidophiles, pH 0 to 4, face a perpetual proton-hydrogen ion influx. Their primary defense is active exclusion. Highly efficient proton pumps continually export hydrogen ion, often using ATP. They often maintain a positive membrane potential to repel cations, effectively sealing the cell. Alkaliphiles, pH 9 to 12, have the opposite problem: proton scarcity. They use sodium and hydrogen ion antiporters to import protons while exporting sodium ion, often coupled to motility or nutrient uptake. Piezophiles, surviving pressure. Piezophiles, barophiles, live under extreme hydrostatic pressure, found at deep-sea trenches, up to 1,100 atmospheres. High pressure compacts molecules, reducing the volume of the system. This decreases membrane fluidity, favoring solidification, and can distort protein structure, inhibiting enzyme activity. Piezophiles adapt by increasing unsaturated fatty acid content, maintaining fluidity, and evolving protein structures that are compact and resistant to pressure-induced dissociation. Why survival occurs? The fundamental question remains: why do they survive where mesophiles cannot? The answer is structural and energetic robustness. A mesophilic protein has evolved for marginal stability. It must be flexible enough to bend during catalysis. If we increase the temperature, we provide enough kinetic energy to cross the activation energy barrier for unfolding denaturation. A thermophilic enzyme has a higher activation energy requirement to unfold, Evolution has tuned its protein-folding landscape, sacrificing some flexibility at lower temperatures to ensure the folded state is deep and thermodynamically stable at high temperatures. In this context, the extreme environment is the optimal environment for these specific molecular architectures. I'm looking at some old notes on Sulfolobus. It's an acidophilic thermoacidophile. It grows well at pH 2 and 80 degrees Celsius. We were trying to purify its RNA polymerase, and I remember writing a note to myself. The conventional purification buffer, pH 7.4, is destroying the protein activity before we can assay it. It's easy to forget that when we study these systems, we are the ones imposing the stress. We pull these organisms from their normal, acidic, hot home into our neutral, chilly lab environment. We call it an extreme environment, but for Sulfolobus, pH 7 is the toxic extreme condition. This is the central challenge in extremophile research. You must design your experimental conditions to mirror the organism's optimal environment, or you aren't studying physiology. You are studying artifacts of death. Common research pitfalls. This brings us to critical troubleshooting when culturing extremophiles. A common pitfall is defining an organism by only one extreme parameter. Consider Acidophiles ferrooxidans. It is a famous acidophile used in biomining, thriving below pH 2.0. If you are struggling to culture it, your reflex might be to increase the temperature. However, A. ferrooxidans is a mesophilic acidophile. It grows best around 30 degrees centigrade. If you incubate it at 60 degrees centigrade, it won't grow. The error lies in assuming that extreme adaptations are always linked. We have thermoacidophiles, but we also have mesophilic acidophiles. Never optimize two variables blindly. Always define the physiological range of your specific isolate. Why do we invest in understanding these mechanisms? The answer is industrial biotechnology. Consider enzyme-based manufacturing. Many industrial processes, like breakdown of biomass, require high heat and organic solvents to solubilize the substrates. Mesophilic enzymes are useless here. They denature instantly. However, the cellulase purified from the hyperthermophilic bacterium Thermophiles maritima remains stable and active at 90 degrees Celsius. These extremozymes offer superior stability, enabling faster reactions, reduced viscosity, and resistance to chemical denaturation. Extremophiles are not just ecological curiosities. They are a library of robust, industrially resilient catalysts. We have covered a lot. To summarize, bacteria are extremophiles because of specific molecular adaptations that manage stability. Thermodynamic stability is achieved in heat via saturated fatty acids and reinforced proteins, cold via unsaturated fatty acids, high salt via the Salton mechanism, accumulating potassium ions to stabilize unique acidic proteomes, high and low pH via precise active ion exclusion, like proton pumps. Survival isn't a miraculous defense. It is a direct consequence of a molecule's evolved optimal energy landscape. Understanding this distinction is vital for both culturing these unique microbes and harvesting their enzymes for industrial application. Thanks for watching. I'll see you in the next session.